Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker-Anthony, Scientific Editor of the Molecular Therapy family of journals. This episode features a conversation between the Editor-in-Chief of Molecular Therapy Nucleic Acids, Dr. Paloma Giangrande, and Dr. Krishanu Saha and Namita Khajanchi from the University of Wisconsin-Madison. They will discuss an article recently published in mTNA by Khajanchi and colleagues titled Controlling CRISPR-Cas9 Genome Editing in Human Cells Using a Molecular Glue Degrader. If you enjoy this episode, you might be interested in our hybrid event this fall that will allow you to engage with pioneering researchers in gene editing. Breakthroughs in targeted in vivo gene editing will be held in San Diego and virtually November 20th to 21st. Visit asgct.org/breakthroughs for more information. We hope to see you there. And now, Dr. Giangrande, Saha, and Khajanchi.
Welcome to today's episode where we explore a breakthrough in gene editing technology designed to fine-tune CRISPR-Cas9 activity and improve the safety for both research and potentially for future therapeutic applications. Our featured study, which is entitled Controlling CRISPR-Cas9 Genome Editing in Human Cells Using a Molecular Glue Degrader, describes an innovative system that uses a miniature Degron tag to degrade Cas9 nuclease on demand with a pomalidomide, a clinically approved drug. So what does this mean for controlling off-target edits, enhancing cellular health, and future directions for genome engineering? Let's discuss these questions with our authors and dig into the experimental data that opens new doors for somatic gene therapies and precision medicine. So welcome and thanks for speaking with us today. Let's start with our first question. Can you tell us what led your team to focus on temporal control of Cas9 activity, and how does the molecular glue degrader approach address major safety concerns around gene editing? So whoever wants to take this one.
Well, thank you, Paloma. I'm really excited to talk about this work. I think our approach was to think through the scenario where Cas9 and gene editing activity doesn't stay on for the entire lifetime of a patient after delivery. Really, the hope here is that it's a one and done type of treatment, because once the genome edit has been made, in theory, that should be durable effects from expression in the genome for the lifetime. And so we've done some previous studies looking at how long do we need genome editor activity to be active for a particular edit to happen. And it's really on the time scale of hours to days in most of our studies before. And so we were looking to establish that type of tight control over time such that we can turn on and off genome editing at that type of time scale. And we knew that if you had too much nuclease activity, you also end up recutting the genome potentially after a double-strand break has been repaired. And multiple rounds of cutting and then repair, cutting and repair may lead to some outcomes that we might not want. And so this is some of the kind of motivation in looking for systems that could, be controllable. And I'm going to hand it off to Namita, who really drove the study of the system, the degrader system, and how she designed it.
Yeah, that'd be great if we can drill into a little bit more of the, the details of how the Cas9 degrader system is constructed or how you constructed it, and specifically how this small molecule, pomalidomide, triggers the degradation in human cells. Yeah, go ahead, Namita. That would be great.
Awesome. Thank you, Paloma. I'm really excited to be here. Thanks, Chris, for answering that first question. So the Cas9 Degron system itself is on a lentiviral construct, specifically a lenti-CRISPR vector. And so basically to put the Degron in, we had to use specific enzymes and make sure that it was on the Cas9 plasmid itself. And so specifically, a Degron is a small chain of amino acids that we were able to fuse to the Cas9. And the idea of it is that once we administer a drug called pomalidomide, or POM for short, once we administer it, POM can now bind to the cell's, like the cell's own degradation machinery, specifically a part called the cereblon. And so this interaction creates a new sort of protein to protein interface. And so once POM is on the cereblon, now POM comes and finds our Degron, which is tagged on our Cas9. And then really that's when the ubiquination starts. So the degradation machinery ubiquinates our Cas9, and then this leads to ubiquination and then subsequent degradation via the proteasome. So in this way, we can really control Cas9 when we want it to basically disappear.
That's great. That's great.
I'll just add that there's a lot of systems out there that can control Cas9 activity, and that could be through transcriptional changes, through anti-CRISPR proteins that kind of inhibit activity. We thought actually getting rid of the protein itself through a lot of really exciting work in the protein degrader fields would lead to a really rapid degradation response. So that's kind of one of the rationales for going to a protein level control. And I think one analogy that we use is that this is like Cas9 with a dimmer switch, in that you may be able to also control the exact level of Cas9 activity you want by controlling the protein level inside a particular cell. Because different cells may need different levels of Cas9 around, they can be degraded, tagged for degradation, and turn over in different ways. It could depend on how you deliver the Cas9 as well, if it's through mRNA, plasmid, AAV, all of those different delivery strategies. You have particular constraints on the exact levels of Cas9 and the distribution of that in a target cell population or tissue. And having this type of kind of orthogonal control to tune or dim Cas9 to the right level, we thought could be quite important.
Yeah, that's great. That's really interesting. And can we talk a little bit more about why you used the Degron that you used? My understanding is that there are several different types of Degrons, and if you can dig in a little bit more into the different types and what they do, the differences and similarities, and why did you land on this lymphoid restricted transcription factor Degron? So yeah, if you can expand on that. Is this like the only Degron that is responsive to POM, or are most Degrons also responsive to POM?
I can take this question. So rather than initially really thinking of the Degron itself, we really wanted to identify small molecules we had three things that we wanted to look for. A, that they're FDA approved. B, that they're really cell permeable, and C, that they can cross the blood-brain barrier. And so for us to be thinking already, like, what is the potential of using these in vivo? So we thought about that. And so the small molecule that came about was pomalidomide. And that was our top candidate because it's also used in treating multiple myeloma. And then we also know it can degrade IKZF1 and also IKZF3, which are a group of transcriptional factors. And then particularly this Degron, which is from IKZF3, it's great because it's very short. So just the one Degron itself is 25 amino acids. After we ended up doing a double Degron, in total, it's only 58 amino acids with the linkers. And one of the really big advantages of using this particular system is we really don't have to co-express anything else exogenous aside from the small molecule itself. And that really helps us simplify both delivery and also reduce any sort of immunogenic risks. Chris, do you have anything else to add?
Yeah. I mean, I think some of the original inspiration for this work was in discussions with a strategy targeting APP for Alzheimer's disease treatment. And this is a collaboration with Dr. Subhajit Roy, who was at Wisconsin, has then moved to University of California, San Diego. And his postdoc and our lab came together and started thinking, well, if we wanted to edit APP in the brains of patients with Alzheimer's disease, we'd really want it to be quite safe. We'd want it to be able to turn it off. And we started thinking about these types of designs. So in some ways, to answer your question, Paloma, how do we come about it? We came about it in the reverse direction. We were thinking about an application in the brain and being able to rapidly turn something off in a reversible manner in patients without it being very risky. And that kind of narrowed our search space down into thalidomide. And, then this work that Namita embarked upon to see, okay, is the machinery there in neurons and, expression databases, et cetera? Is it on in other cells that, maybe targets of in vivo somatic cell genome editing? Those are some of the questions we answered. And in many ways, I think the answer is yes. This Degron system seems to work quite robustly in many of the cell types that we tested in the paper.
Yeah, and I guess one question I have, which is specific to POM, the small molecule that, it's been clinically, approved. It's a class of immunomodulatory drugs, and my understanding is that it's been found that to have numerous, for example, off-target neosubstrates, which are other proteins that can also be degraded by this drug via similar mechanisms. Can you touch on potential off-target and on-target effects of the system, in particular as it relates to your Cas9 system?
Yes, it's certainly a super important question. We don't want to mess up the activity and precision of Cas9 by adding this type of tag. And so we did a number of studies with various guide RNAs looking at different targets and various cell types in the paper. And we saw pretty robust on-target activity that could be controlled even when you add the Degron. So the overall activity of Cas9 didn't really seem to be that significantly affected. And then the indel profiles upon DNA repair of the double-strand break look also pretty similar. So I don't think we're majorly changing too much of what the precision of the type of edits that we make. We looked also at predicted off-target sites. Mm-hmm. And those assays were actually pretty challenging because our guides in many ways didn't have very high off-target activity. So the signal that we would look for in the off-target site wasn't, very high. That said, I think what we found, which was in some ways to be expected, that if you lower the protein levels, we get reductions at both the on-target and the off-target sites. The ratio of which I think in some cases seem to be favorable in that we get a little bit more on target activity relative to off when we kind of turn it off rapidly. But in many cases, it was kind of the same, the on to off target ratio. So that was actually a hypothesis that Namita explored, and to see whether would we get better precision by controlling Cas9 at this level. I think the answer is largely no, because we didn't see a huge effect in terms of precision. I do think what we did see is, certainly that we could control the activity robustly and reversibly. And that's why the dimmer analogy might be a better analogy than, having a better arrow to hit a target.
But I think that's really encouraging, because you're not seeing, more off target with your system or with the addition of POM. So I think that's really encouraging.
Yeah. I mean, I would say the most stringent assay we had was in the induced pluripotent stem cell derived neurons, human ones, where I know those cell types are really sensitive to all sorts of compounds and perturbations. We've infected them with lenti-CRISPR and other things before, and some of them are really sensitive and they die. And so we looked pretty carefully at what would happen when we had the Degron system in there, and with the appropriate controls without the Degron, and then adding POM at various levels and not. And, we saw largely great function of those iPSC-derived neurons. And so electrophysiological assays also seem to confirm that. So to me, that's pretty encouraging that the system, seems to function well without much genomic off-target, but also kind of physiological off-target effects in a human neuron.
Yeah. Yeah, no, that's fantastic. And you said that, you started, really thinking about this from really the end. It's like, where do you want to get to, and the application, which is in, Alzheimer's, going after APP, so targeting neurons. Can you talk a little bit about when you've compared the, especially the editing efficiencies, but also some of the specificity of on-target effects in other cell types? Like, is this system transferable to potential other indications, other tissues, other types of diseases? Like, do you notice a similar pattern, or is this system more really specific for going after CNS type indications?
I can take a part of that question. So we tested this in HEK cells, but most particularly the more important ones, stem cells, hepatic cell lines, and neurons. And we saw that with our POM addition, we are seeing like that decrease in editing overall, all of those cell lines. So it's very transferable in different cell lines. Whether or not the editing activity is the same differs among the cell lines. And then that relates back to us thinking more about the neosubstrates. Some cell lines express some of those neosubstrates even more. So now our pomalidomide might be, degrading those instead of really our Cas9 at that moment. And so this is transferable over different cell lines. And that's why we really looked at a range of cells from different places.
Yeah, I think it's a great question. And we're hoping that the field kind of takes it on, because, of course, these limited cell types that we looked at is not the whole set of targets that the field is considering. We see this as a proof of kind of concept study in these cell types that suggests that it should work in other cell types. These questions of whether the degradation machinery is expressed at the right level, whether there's neosubstrates that may also be affected by POM are relevant. In terms of the actual activity of the SBCAS system, for us, we think, based on that set of studies and assays at the genomic level and the DNA repair level, that seems to be pretty conserved across the cell types we looked at.
Yeah. So basically, what I'm taking away from that is that you feel strongly that should not be a challenge. It's definitely a technology and approach that's transferable, can potentially be applied to many different types of diseases and indications. And really, we're back to the biggest bottleneck, as for every other therapeutic modality that I'm aware of, is delivery. If we can deliver it to the right tissue, this can work. And yeah, and we're all, those of us who are working in the delivery space, we're all really, working really hard to overcome some of these challenges. And to date, we are seeing targeted LNPs that seem to be able to deliver large cargoes to many different cell types, in addition to, to the usual liver targeting or hepatic targeting. So I guess I have more of maybe a question for Namita, because we all understand that science is not perfect, and that it can be a little bit challenging to get experiments to work. Can you talk a little bit about what made it challenging in your system to achieve complete elimination of the Cas9 degron protein via POM? In order to abolish that nuclease activity on demand, and what you consider, and would you consider this like a surmountable technical hurdle, or does it reflect a more broader limitation of the platform, meaning that we're still probably several years away from maybe translating this into the clinic?
Oh, this is 100% like a surmountable hurdle.
Great.
In our current system, we're really using a bit of overexpressing lentivirus in some of our systems. And so there's multiple copies of our Cas9 degron that's being expressed. So, the easiest and best way to go about this is really targeting a single copy. And this, I guess, a lot of people do know, is to insert into like the AAVS1 gene and the harbor locus to really get uniform degradation. And so we would be able to hopefully fully suppress Cas9 activity using that method. So, Chris, do you have anything else to add? I believe, yeah, we'll be able to use this in different tissue types as long as the promoter is right as well.
Yeah, I think we have a number of dials we can turn to try to match the expression level to the degradation level. To us, that's what took quite a bit of time, right, Namita? And I think if we look forward into somatic cell applications in patients, for instance, the eye that we're working on, or in the liver that you heard about from the baby KJ story, there is a distribution of levels of payload that are delivered, even upon a single infusion of a very well-characterized drug product. And so I think I'm optimistic. It still could be quite challenging in a patient that, may have parts of a tissue that turn over the payload, or process it differently, or express it differently. But this gives us another kind of tool in our arsenal to maybe turn off activity when we see some sort of concerning signal. For instance, probably the place where it is most helpful to have this type of dimmer switch is with viral delivery, because there, after delivery is in many cases pretty hard to control the activity of the enzyme.
Yeah, that's great. And I know you touched on this a little bit before, but just heading into like broader implications of this technology, what do you believe are the prospects for adapting this technology, especially for the, as you mentioned, diseases like for, the CNS in particular, liver diseases, or although we have many more therapeutic, modalities that are going into liver these days. Especially with regards to how quickly your system seems to enable, reversibility, and how biocompatible it can be. So if you can just expand on the vision towards the clinic a bit more, that'd be great.
There's so much excitement in my my mind about this. We are working on some non-viral delivery strategies in the eye. And in one of those cases, we have a dominant mutation in a disease called Best disease, where it's only a single base change between the pathogenic allele and the wild type allele. And there's a pretty stringent design requirement for the editor. You have to be able to distinguish really one base. And it can be done, but the relevant off target there is the wild type allele. And that's where, we want to make sure that our off target in that case is really untouched. And it might be that we need to turn off the editor really quickly to get that type of precision. So it's hard to know exactly at the moment, based on where we are with these types of studies, what applications this may have the most impact in. But I could imagine scenarios like that one in the brain as well, where this would have quite a bit of impact, because again, there's also these types of dominant mutations in the brain, a place where you might have to use a viral vector to get enough on-target efficiency, but then you want to turn it off quickly. And in terms of biocompatibility, we've chosen already the inducer to be very, compatible in terms that is FDA approved at pretty high doses. One of the challenges there is that are the doses that are considered safe through the studies and with multiple myeloma patients, does it get to the right level in all parts of the brain to turn it off uniformly? Our thought is yes, although we haven't done that study obviously yet, and that's something that should be carefully considered. And then the liver is also an interesting place, because that is also a natural place where drugs, build up and are excreted. So I don't know, Namita, you've looked at some of these targets in the liver as well.
Yeah, I was gonna just add just a little bit more. Like in our hepatic cell lines, we actually saw the lowest amount of editing, and we think, there might be a reason for it. Potentially, it's getting metabolized in the liver cells quite quickly, like the the pomalidomide itself.
Yeah. That makes sense. Yeah.
So that's something we might look forward to, studying, figuring out, because one of the specific genes that is really looked at in the liver is the PCSK9 gene, and that's related to your cholesterol levels. And so, once again, if we can edit PCSK9 and get rid of Cas9 as quick as possible, like that's our goal, is not to have prolonged editing.
Yeah, and I would say with that in mind, like, is there a way that you could co-deliver the POM in a way, or deliver the POM specifically, let's say either to CNS or to another target tissue of interest, and not deliver it systemically? So in a way to increase the local concentration and also potentially avoid some undesired off-target effects in other tissues.
Fantastic. Yes, I totally think that's a way to get around some of the systemic concerns of POM application. And there are all of the innovations in terms of drug delivery with a small molecule now is able to control genome editing. So that's some of the exciting, I think, possibilities that we get by controlling Cas9 activity this way. You could co-deliver it to keep it off, and then as POM goes away, it turns on. That's also a scenario, perhaps, in particular clinical application, just thinking out loud here, where you do a surgery, a resection for a cancer, you put in both the editor and POM. It doesn't edit right away, but in the remaining cells, as they perhaps residual cancer cells that are around, this turns on and it edit something that you'd like. So this is a set of technologies that I think hopefully, in my mind, opens up some applications that clinicians and the greater community come to us and say, look, this might be a cool way to control activity temporarily that is really clinically relevant.
Yeah, that's great. And I guess maybe let's just talk a little bit about how your approach maybe differs, or what does it add to other Cas9 control methods out there, like, split Cas9 or other inducible systems, either with light or small molecules. Is your approach more amenable to enhanced safety and precision compared to some of these, more translatable into the clinic? What are your thoughts on that?
So I think this is a really big question because there's both disadvantages and advantages to all of the systems that exist. So far, sometimes it's related to size. And then with the split Cas9, for example, we have to make sure both of the Cas9 components are able to go into the nucleus, et cetera, at the same time. And then most particularly with the light, it's whether or not that light can penetrate through tissue that we would want, or in this case, body, if we're thinking in vivo. And so the small molecule method is very, great. But if we do combine it with some of these other methods, because there is light that can penetrate to the body, through the body, for example, we would really have even stricter control over our Cas9. And something to think about is having an on and an off switch. Right now we have that off switch, attaching an on switch, right? Which could be any one of these control methods would be great, because we would get even more safety in that effect, and we would get more temporal control.
That's beautiful. Yeah.
I was going to say that we can engineer all sorts of cool controls, and I think these are great ideas, and it really may be the kind of precision we need in the clinic. We've come, one of the real advantages I see of this technology is that it really only requires delivery of one entity, which is in terms of getting the system set up. Some of the split systems requires dual AVs, or the anti-CRISPR requires sometimes another vector or a larger payload to be put in. This amount that we're adding in using the Degron is pretty small. And in that ways, I think addresses the central point that you brought up, Paloma, of delivery, that in itself, at least we're not adding additional delivery challenges when we add these dimmer switches and controls. So I like simple strategies, and in some ways we don't want to add an additional delivery headache for the field by adding, maybe a control switch that is not that needed in some scenarios.
Yeah, I can't agree more. I love that too. Simple, usually the simplest solution, usually the best solution, and sometimes over engineering can work against you. Yeah, that's fantastic. Do you see that this Degron system can be used to regulate other genome editors, like base editors and finger nucleases, or other transcriptional regulators?
Yes, I think the principle is very similar. We tune protein levels, which should change activity. So I know there's some work out in the field that have started exploring that, and I think they see pretty consistent results with that type of hypothesis. I don't see really much fundamental barriers to attaching this Degron, again, because it's kind of at the end of the enzyme and pretty small. And so the fact that it might interfere with something kind of outside of where the R-loop formation and other kind of key enzymatic steps are happening in terms of genome editing typically should not happen. Epigenetic editors, you could probably put it at different parts of the fused epi editors. I think there's lots of possibilities there.
Yeah, that's great. No, and this has been fantastic. Lots of great ideas. So maybe let's wrap up with one last question. Where do you really see additional work being needed before deploying this system in clinical gene therapy trials? And also, if you want to touch on what types of diseases you think are going to benefit the most and are going to be the first ones that are really going to, you're going to enter the clinic with.
I think in terms of additional work, we're really hoping that other researchers are interested in, taking this technology and trying it out in their own different labs and their own perspective. In terms of clinical gene therapies, I think right now we really have this as a proof, right, that this technology works. And taking it in vivo, at least, starting with mice will be super easy. I know they're changing a bunch of stuff now as to, whether or not it should be animal tested or not. But I think Chris can really talk more about the clinical part because I've only been an in vitro scientist thus far.
We're certainly excited to explore retinal applications. We have our CRISPR Vision program supported by the NIH Somatic Cell Genome Editing Consortium that's looking to look at a number of pathogenic alleles. And then some of them require high precision, and then some of them are in cell types where, perhaps the lipid nanoparticle can't get into, like photoreceptors very efficiently. There, we could go to a viral vector, potentially with a Degron system, to be able to establish great levels of control. And then also the brain applications. I know a lot of folks in our consortium are excited about moving some of the new delivery strategies that can cross the blood-brain barrier to get a vector in, but then you also want to perhaps turn it off, the activity. I that as being very exciting. I mean, I agree with Namita that there's quite a bit of work that needs to be done to move this into first-in-human trials, but I do think the community has a lot of momentum on its side at the moment, given some of the advances in delivery vectors, in these control systems, such that we can get genome editing to be as precise as we need to have safe and efficacious drugs.
Yeah, no, that's great. And I hope that you're getting a lot of interest from, biotech and, companies reaching out. Because I think that work can move a lot faster, in collaboration with, biotech companies. But this has been fantastic. I want to thank you again for your time and for, discussing your work with us. Thank you so much.
This was a really great discussion. Thank you.
Thank you, Paloma. Very excited to just share my research and what I've done over the last couple of years.